stratifying in an intensive care setting.
Exactly, thank you. It's a pleasure to talk to you. And yes, I got a research professorship from the NIHR this year, which is around precision medicine in sepsis. I'm also one of the directors of research for the Intensive Care Foundation, so running UK multi-centre trials within the ICU. And so, for those of you who don't know, sepsis is defined as life-threatening organ dysfunction caused by a dysregulated host response to infection. So you can already see it's a very heterogeneous syndrome that we're talking about, hence the need for stratification. And it can affect anybody. Recent estimates suggested there could be as many as 27 million cases throughout the world and 8 million deaths from sepsis. If you concentrate in the UK, for those patients who come to intensive care units, the really severe patients, over 100,000 of those every year, and 37,000 of them will die within a few weeks of presentation. And those rates are rising as the population gets older and as we treat more and more complicated diseases, the numbers are going up each year. And we know that the key to good outcomes is prompt treatment. And we're talking hours here, sometimes minutes, but generally hours, but not the days and weeks, maybe other conditions. And our treatments, some that really quite simplistic, I put them down here, are treatments that takes you 7 years to train in to become intensive care. But you have to resuscitate the patient, give them oxygen and fluids, antibiotics, drugs to help the blood pressure, so vasoconstrict or to increase contractility of the heart. You need to support the other organs, mechanical ventilation, maybe dialysis. And as I said, it's a dysregulated immune response, so we would give immune modulating drugs, wouldn't we? Well, we've been looking for that, and this is a list of all the sort of drugs that have been tried in preclinical sepsis studies. Not all of them have worked, so of course it's not surprising that we don't have a long list like this to treat them clinically. But here I've listed all of the immune modulating drugs that we currently use in sepsis. It's not for want of trying that we've been looking for them. And what has been, we've thought, well, sepsis is an inflammatory response, so therefore it's the hyper-inflammatory patients who do particularly badly. So if we block that, we'll reduce the deaths and patients will do better. So inflammatory cytokines are important. TNF, tumor necrosis factor, blocking that in other inflammatory conditions, rheumatoid arthritis, Crohn's disease, works. So again, it was tried in sepsis. We can see here some of the early studies using a soluble receptor to sort of mop up the TNF. The low doses just didn't work, and the higher doses actually made patients work. So it wasn't going to be quite so simple as just blocking inflammation. So different drugs were tried. This was a monoclonal antibody to TNF. There are multiple studies. I'm just going to show you the one. But you can see there was no effect on outcome. So it was thought quite a long time ago, well, obviously what we need to do is stratify the patients, and so these trials were attempted. It used another inflammatory cytokine as a stratification variable using interleukin-6, so another key inflammatory cytokine. It was a simple qualitative test where you're above or below a certain threshold. And you can see that it works quite good, quite well as a prognostic stratification. If you're IL-6 negative, you have a better outcome than if you're IL-6 positive. But you can see in this trial, it was only the IL-6 positive patients who were randomized to the treatment of the placebo, and again, it still didn't make any difference to outcome. And this has been tried for a number of different molecules, different drugs, and none of them were made it into long-term clinical practice. For a while, we did have one drug, activated protein C, Zygris, made by Eli Lilly, that after the Prowess trial in 2001 got a license for treatment. The trial was stopped early at an interim analysis because of a significant reduction in mortality. Now, there was a lot of controversy about the drug and the trial, some of it fair, some of it I think very unfair. But I won't go through all of it, but it led to Eli Lilly having to run a second trial 10 years later, and that came out with no difference. Eli Lilly then withdrew it from the market. Now, this was a drug that personally I thought in some patients did seem to work extremely well, but we no longer have it available to us. While I was aware of all these problems with this, I was doing a postdoc in Vancouver at the time, and with my colleagues there, we realized that we needed to stratify the patients, and we tried to develop a pharmacogenetic test around response to APC. We basically used an approach where we had two genes here, and I'll just say if you had the responsive genotype in both, you were plus minus if only one, and minus if you had none. You can see in this was a case control study, matched controls we found. We thought an interesting result. We persuaded Lilly to look at it in their data, and they found the same result. This was from their RCT. We did another multi-center observational cohort study as well, where we saw the same pattern. One of the issues, of course, this isn't statistically significant with the interactions, but we went to the FDA to ask them how would we take this forward if we wanted to develop a diagnostic. And I was disappointed at the time. I didn't know about the various trial designs, but their only answer was we need to do a trial that's powered enough to test for an interaction, which was clearly impossible. I would have said even for big pharma to run such a massive trial, the numbers would have been required. But anyway, the drug was withdrawn, so we never got to fully develop the diagnostic. So hopefully I've persuaded you why we need stratified medicine. It's not just my own personal thought. This commission paper in Lancet Infectious Diseases last year from a number of leading sepsis researchers pointed out the problems that it's a heterogeneous syndrome. The single biomarkers are probably not going to capture the complex changes. That even if we measure things in the circulation, they're not necessarily capturing what's going on at cellular level. And our current tests that we use, particularly our microbiology tests, are just too slow. So what could we do? What could we stratify? Well, I've listed here, highlighted in blue, that part of the treatment where I think there's potential. Basically, it's around all of the drug treatments. I'll start with the cardiovascular drugs, the vasopressors and the inotropes, because that's the two trials that I've been running in the UK in the last few years. And what I'm going to be testing within this professorship. There are a number of, so with pharmacogenetics, there are a number of polymorphisms in various adrenergic and vasopressin pathways already well described. They're showing vascular reactivity, varies response to catecholamines in terms of blood pressure response, heart rate, even potentially sudden cardiac deaths have all been associated with these genotypes. There have been some preliminary studies in sepsis, again suggesting these polymorphisms associated with mortality, serious adverse events. And so we hope to sort of validate or refute these in my trials, so sort of validation cohorts. But how would we exactly stratify? Well, it could be around just even what blood pressure targets we aim for. We're still not sure what it should be. Sort of evidence is that some patients, particularly if they're hypertensive, if they have a higher blood pressure target that we aim to restore, we may reduce rates of renal failure, but equally then we know we get the adverse effects from the drug, so could we stratify there? But could it be around our choice of vasopressor? So we generally use as a first-line treatment the catecholamines, adrenergic agents, but again we know they have side effects. And there are a number of sort of non-catecholamine alternatives. So vasopressin is the one I've been testing, steroids. Do we add in inotropes, not vasoconstrictors, to actually increase the contractility of the heart? So this was my vasopressin trial that we published a few months ago. And you can see we were interested whether the sort of suggestion that vasopressin may help maintain perfusion of the kidney. And we did see less requirement for dialysis in those who were treated with the vasopressin. Unfortunately, sort of counter that, when we looked at the adverse events, there was more digital ischemia in the vasopressin treated patients. So again, could we use the genotype to potentially stratify which patients were going to get the vasopressin or not? The other trial just published last month, levosimendan, inotrope, to basically, could we improve organ function by improving cardiac output and regional blood flow? What we did see, didn't show any difference overall, was there was this increased heart rate with the patients that were treated with the inotrope. And we know that high heart rates are not a good thing in sepsis. And again, when we looked at adverse events, there was more tachyarrhythmias in the patients who were treated with the drug. And so this is and other evidence, again, suggesting actually, do we need to slow people's heart rates down? Do we need to beta block them, which is sort of contrary to all of the treatments we've always given in sepsis. So again, could we identify those patients who are going to benefit most? Next bit is around antibiotics. What could we do there? And as Munir mentioned, around point of care testing, this is vital for what we do. There are lots of new devices being developed. I've just mentioned the one that I'm working with. It's a spin out from Imperial College, from Chris Tumazu, where he's using pH sensing semiconductor systems. You can do gene sequencing on little chips like this. What they're actually developing at the moment is for sequencing bacterial DNA, and they claim that within the next 12, 18 months or so, they'll have a device where we can actually get the sequence. There's about antimicrobial resistance in some cases in 2 to 3 hours compared to the 2 to 3 days that we wait at the moment. Now, it's perhaps not what we might think about stratified medicine, but it certainly will help us improve our antibiotic prescribing, which hopefully will reduce antimicrobial resistance, which is a major problem. But maybe we can stratify patients according to the type of their infection. And the other thing is the technologies can be adapted, so we could use this for other testing, so rapid testing. But maybe knowing the type of infection actually is a form of stratification. So this is work that I've been doing for a number of years with Charles Hines, Adrian Hill, and others at Oxford. And we've got doing a genome-wide association studies in sepsis. When we looked at all sepsis, we didn't really find any hits. There's these Manhattan plots suggesting no clear signals coming out about genetic responses and outcome in sepsis. When we narrowed it down to the predefined, we had already planned to look at community-acquired pneumonia. We did start getting a significant hit. We got one here. And importantly, when we looked at a number of different cohorts and studies from around the world, we actually got a consistent signal, but we only saw it when we looked at the community-acquired pneumonia patients. So we did have to bring it down to that subphenotype. It wasn't significant, but when we even got down to those where we had proven bacterial infection, and particularly we went with those with the streptococcal infection, strep pneumoniae, we got an even stronger signal. Of course, the numbers now, it's not significant, but it was encouraging. So actually, maybe we do need to get down to individual infections. And then finally, could this be the answer for developing immune-modulating drugs? So again, this is work I've been doing in collaboration with Julian Knight, who's been leading it at Oxford, where we've taken these same patients, but we also collected their gene expression profiles from the blood. And we used an unsupervised hierarchical cluster analysis, so identify natural groupings within the data. And we found these two groups, which we called SRS1 and 2, sepsis response signature, 40 and 60% of the population. So a natural grouping within, according to their gene expression profiles. And of the 3,000 genes that we saw differentially expressed when we did a pathway analysis, it wasn't around the inflammatory cytokines where the big separation came. It was actually around immune suppression. And this SRS1 phenotype was very immune suppressed. Importantly, we can actually predict membership of these two groups with just 7 genes, with a pretty good classification rate. And importantly, these natural groupings that we found were associated with outcome, both in the original derivation cohort where we identified them, and then we carried on recruited more patients, and we saw the same thing in a validation cohort. So in other words, could we identify a group here that could be responsive to different treatment, and it seems to be around immune suppression. So I think it's changed our thinking about sepsis. That certainly in some cases we do get hyperinflammatory response, and this may account for some very early deaths. But in many cases, actually, what the problem may be, we can get them over the initial illness, but they're immune suppressed, and further bacterial or even viral infections is causing the problems and the deaths from there. Or in fact, does it change over time, and we need to monitor them over time? These are the things we're looking into now. But it's certainly raised the idea that it's not all around inflammation, and maybe this immune suppression is a new target for treatment and things that we should develop. So my conclusion, I would say, I think that a stratified medicine approach in sepsis is essential for future drug development. It's unlikely to be single biomarkers. Maybe something around the pharmacogenetics may be in that way, but we need, I do think the omics here are helping us discover the biomarkers that we need. absolutely we need rapid diagnostics. I think the technologies are there or just about there that we will be able to use. I think there are issues around regulation. Others have mentioned this. What level of evidence will you need before this comes into clinical practice? One thing I've been able to do is get sample collection along our RCTs that we've been going along, and I think this allows us to collect larger sort of biobanks, and if there was more ways of collecting samples running alongside RCTs, that we can look for this treatment phenotype interaction. Clearly, as we've all said, it's going to involve larger trials. I'll put it in inverted commas because obviously it's larger screening, maybe not such larger numbers treated. I think the UK intensive care community is in a place to deliver on this now, but absolutely crucial to that, and what's made it success in recent years is the NIHR network support. Just to illustrate that, and it's not rocket science, this was the lever-Semenden trial. Our original plan was to recruit over 2.5 years. We got extra funding, we were able to open more centres, it's not rocket science, and we finished actually 6 months ahead of time. So we do need that support, and when it comes in, we can recruit. And it's not just the big teaching hospitals. It pains me to point out that UCH were the biggest recruiter, as I work at Imperial. But more importantly, what I put up here is the other hospitals that were big recruiters. So size relates to how many you recruited. Antrim, Poole. I was at NIHR senior investigators meeting this week, and they apparently the hardest job in the NHS to recruit to is emergency medicine in Hull, I was told. So nothing against Hull, someone else at that. But in intensive care, we were making it work. So even in Hull, they recruited significant numbers. So it is the DGHs, but it does require on the support. And it does concern me as we get to more financially constrained times, perhaps. People have raised up that it's very much more resource intensive to recruit to our trials than perhaps outpatient clinics. And if it's just simple metrics of numbers, if some sites go that way, then we could suffer in the future. I'll stop there and happy to take any questions. Thank you.
Thanks very much, Tony. Time for 1 or 2 questions, Tony. So, can I just start off? We know about, particularly when you're working in an intensive care setting, consent can be quite difficult in terms of getting patients involved in particular studies. Does undertaking a stratified medicine trial add an extra layer of complexity in the consenting process? How do you approach that with relatives and so on?
Yeah, I mean, consent is an issue. I think we've solved it now. We've got mechanisms that work, and it does usually involve speaking to the families at the time and then going back to patients afterwards. I'm not sure that it makes a big difference to stratification, but certainly that's one of the things that I'm exploring in the research professorship is working with patient groups around that. Some of it was also around diagnostic accuracy. So in other words, sort of trading off if you can get the microbiology result earlier, but it's not what level of accuracy would you accept to make a decision based on that? So we were thinking about that. So I'm not sure it's an issue for consent, but there may be issues, other issues around the testing.
Any other questions? Can I ask another one then? Since nobody else wants to ask questions, I might as well. So it's crucial that you are able to recruit through many different ICUs throughout the country, because the patients might be quite rare in terms of the types of patients you want to recruit sometimes. So in terms of quality of data collection, does that vary from the different ICUs and the expertise in recruiting?
So absolutely it requires trained nurses. So the CRN support, we find we can't just use the bank of nurses who work in the hospital and maybe go to clinics and collect it. They usually have had to come from an ICU background. So most of our CRN funded nurses were once ICU clinical nurses. Some of them even work part-time in some hospitals. They like that approach. But these are CTIMPs. We've had electronic data capture already. That was part of the trial. We trained the sites up around it. We had monitors to ensure data quality and so on. You can always find room to improve. But I think the data quality was good. So using the electronic capture helps. There's all the automatic data queries help ensure you have good quality data.
Great. Very quick question.
we go. I think one of the fundamental problems that we've come across with trying to recruit patients into stratified medicine research has been the issue of rejection. And I think, Munir, you talked about this before, and Andrew too, that actually if you have a trial design which stratifies patients to different groups where everybody who you approach then takes part in your research, that's a very different issue for patients compared with a trial where you're selecting patients and rejecting the rest. So I just wonder, and I mean, I'm personally involved in Focus 4, which is one of these sort of stratified platform enrichment trials, but everybody gets randomized, everybody gets into the trial. I just wonder, looking at the data you've just been showing us, whether you see the next stage going forward, you're going to effectively have a stratified design that will be able to offer something to everybody on both sides of the biomarker divide.
Yeah, I don't know what that design is going to look like at this stage. That's sort of what we hope to discover and do. But I can see that as an issue. I mean, we do certainly get that when people got life-threatening illnesses, and we've talked about trials before. You do sometimes get the families go, oh, but I want to make sure they're in the intervention arm, and so on. So I mean, I think that's just about we have to be open and honest with them and try and explain it up front and see if they understand. But I see that could be a problem. I don't know whether that will be our design or not. So but it's something I think we should definitely think about.
Okay, very quickly then, John. Sorry.
I guess the question I've got to ask is the culture in the ICUs in the UK and the culture of the patients different from other countries? Because you hear stories in America that it's very difficult to run some trials when they're not getting the intervention. Is that the culture different amongst our patients?
I've not worked in the States, so I don't know specifically. Things work quite. We have a good relationship with our families and so on. I would say it works and so on. So it's not been an issue. I don't see that as a problem. I would say I definitely get the feeling that most patients and relatives like being in the trial. So one of the trials we had to actually do, we had permission to do it without consent because we had such a short time frame, and we were nervous about doing it. And actually, and sometimes people said, well, I wouldn't have put that patient in. The family are very irate about something, and actually when we put them in anyway, and I went and spoke to them afterwards, and they were so grateful. They said, thankfully, they saw this as a plus. You're caring so much, you're even considering them for trials and so on. So actually it was a plus putting them in. So I think that patients do like being, and their families like being part of the trials.
Thank you very much, Tony. That's great.